Hi everyone, welcome to Foresight's Molecular Machines group. I'm really happy to see so many of you here. Happy New Year. I wasn't quite sure how long you can actually still say this, but I was just being told that if you hold someone dearly, you can say this, and for sure that's true for this community. So I'm continuing to say it, perhaps until January is over. But yeah, thanks a lot for joining us again this year. It's now, I think, the third iteration of this group. Really happy to have Basile here today to discuss a little bit about the work that some of you have missed when you were at the Molecular Machines conference. But just to give you a little bit of an understanding, we had our Molecular Machines conference last year. Many of you were already there. If you guys ever want to read up on the report is out, but we also now have all the videos and video recordings on the page. And unfortunately, Basile couldn't make it at that point, but he sent the wonderful Sinu instead, who did a really great job at discussing the work. So if you're ever interested in like a bit of another deep dive, probably some of it is already outdated, but she did a really wonderful job. So all the videos are on there now. If you want to know more, that's there. I also do want to make you aware that we have an upcoming workshop as well. It's the same shtick. This time we're diving deeper into different design tools for molecular machines and are trying to put some pieces together already. We have a few wonderful folks already confirmed. And if you're interested in applying to this, the applications are already open on the website. I'm going to share them here in the chat. If you're applying from within the seminar group, then just mention that you're part of the group. And the applications are open to outside, to external folks as well. Okay, cool. Well, that's, I think, most of it that I wanted to start with. I think I'm going to give you the stage now. I have tons of questions. There were a lot of really interesting Twitter threads that were started after Sinu's presentation on your work, after the workshop. And so we have a lot to dig into. And thanks also for providing a challenge and an abstract of your talk. So I think we're really well prepped for the afterwards discussion. But for now, I'm really delighted to have you on here. So thanks a lot, Basile Wicky, for joining you from the Baker Lab, and you'll be discussing designing de novo interactomes for biomolecular computations. The stage is yours.
So I'm very happy to be here and obviously to present this work on what the title sort of reveals, both encompassing aspects of protein design and aspects of hopefully sort of synthetic biology and biomolecular computation. All right, so hopefully this is now live. Okay. So perhaps to start, I will just give a very quick foray into protein design, which will provide the backbone for what most of the rest of my talk will be about. And as a very quick glimpse, the protein design field has evolved tremendously over the last sort of 2 decades, from relatively small, humble beginning in '97, where the first computationally designed proteins sort of were verified, to sort of iteratively more complex structures, first bundles, then the first, the Novo fold with top7 in 2003, and then sort of gradually increasing in complexity, going into larger proteins, symmetries, alpha beta structures, and so on. And all the way to, in the recent years, things like molecular motors by Alexis Coubert, who spoke at that seminar before, or bioswitches by Lajoie et al. Now, what this, this is tremendously exciting, obviously. I sort of stopped at around the 2020, 2022 mark, because, of course, I'm sure all of you are aware how AI and machine learning are, really revolutionizing the life sciences, and it is true for protein design too. So I will talk about this a little bit in the second part, but that really has led to a space transition in terms of what we can achieve. And this is really exciting because what some of these proteins used to take perhaps something like 5 years for a single postdoc to get to that stage, we can now do in the dozens at a rate and a scale that is just really unprecedented. And so I think we're really poised to enter a new era where we can move into sort of systems biology or more complex system. And that's what I'm going to talk about today. But perhaps, how very briefly as an overview, how do we do protein design in the first place, right? So we always aim for sort of a function often. And if we do that through structure, we sort of separate the problem in 3 stages. The first, we define that problem in geometric terms. So maybe it is designing for specific symmetry we're interested in, or we want to target a protein surface on a natural receptor, some of the work that Jane represented at the workshop. or perhaps binding a small molecule or an enzyme, in which case we need to define a pocket and a enzyme. So once we've defined that problem geometrically, the next stage is to generate viable backbone geometries. By that I mean obviously a trace and just the backbone of the protein that would accommodate the shape so that we can actually generate hopefully the function we're interested in. And here that second stage is where already machine learning has had tremendous impact in terms of our generative abilities to explore space. And then in the last step, obviously, we don't encode XYZ coordinates, we encode amino acids, and that obviously through the form of DNA. So we do need to give those structures a sequence that guarantees that we fold to that structure. And so that last step is basically sequence design. Now, there's been traditionally in the most of the slide I presented earlier, actually all of it, was done in this idea of energy-based protein design, which is rooted in sort of molecular mechanics. So you have a shape and you search in sequence space through rotameric combinatorics to try to minimize the energy of that structure. So if you think of this sort of abstract energy landscape representation, you have your target structure and you want to sort of dive the energy landscape to ensure that you get there. So this is just saying that you minimize the energy given the structure. But one of the reasons why protein design was always very difficult is by doing this, as you can see in this illustration, you always have the risk of generating alternative conformation that you're not aware of because you sort of narrowly focus on that target structure and nothing else. But of course, there's been tremendous advances in deep learning applications to structural biology, where very simply put, you put a sequence, you pass it through a network, and you get a structure. But really what you get before getting the structure, which is sort of collapsing what the networks understand in probabilistic terms to a single X, Y, Z set of coordinates, you actually have a whole set of distributions, a probability distribution and ij distance between all the pairs. And that's pretty valuable because that means that now, instead of having this sort of deterministic single structure, a single energy point, a scalar value, you have a probabilistic mapping between a sequence and a structure. And that allows us to hopefully generate a more sort of holistic view of the energy landscape for proteins so that we can sort of guarantee that in the design process, we sort of target the protein the structure we're interested in, while at the same time ensuring that we have no alternative states. In other words, we are trying to maximize the probability of the structure given the sequence. And these advances in the last sort of 2 years have really made protein design much more efficient, which, as I mentioned, is really instrumental, I think, in how we can move forward into more systems biology. Okay, that was my very quick introduction to protein design. I'm happy to talk more about these topics in the discussion afterwards. So now moving on to the purpose of my talk, which is the Novo Interactomes. So obviously, this is located in this idea of biomolecular computation, and perhaps the most striking example of this is gene circuits, right? So for the last 2 decades, the community has tried and strived to build systems that can compute within cells using gene circuits. And I'm using this singular example not to say that there's a lot of other exciting work that has been done through the years, but this is sort of the culmination of this idea of, can we program living organisms the way we program a computer? And so the framework and the thinking is to say, well, we define a function, a Boolean function, we sort of implement that in sort of classic circuit design, like electrical engineering, where you have individual Boolean gates that cascade into each other. And then we sort of translate that into a circuit where a set of repressors and activators of genes sort of laid to that function. And this is the work by the Voigt Lab in 2016, the cello implementation, which really demonstrated that, it goes a long way and can achieve a lot. Now, this particular set framework of biomolecular computation is sort of emblematic to the field, which is to say it does all the computation at the transcriptional level and uses a framework that relies exclusively on orthogonal components. And that's important for composability because you want to make sure that you don't cross over, because otherwise it's very hard to imagine what you do. And this framework is now been more recently implemented in the case of proteins, and those are 2 recent works, one by the Ellos Lab and one by our lab, where using the similar framework, if you can have protein components that are orthogonal to each other, in the case of the left, protease based, in the case of the right, protein interaction based, you can implement functions. Now, this is obviously exciting, but there is actually, first of all, very little work that has been done on the protein level, despite obviously nature doing a ton of computation in protein substrates, right? I mean, we can think of all the signaling pathways, all the enzymes and kinases, etc. So nature does not only operate at the genetic level when it performs and processes information, but perhaps more importantly, nature doesn't really, if we think of it at, if we open a classic paper or a textbook in molecular and cellular molecular biology, it doesn't really entirely rely on orthogonality, and quite the opposite. So here I've just selected two examples that I'm particularly fond of, but there's obviously an enormous literature out there. But there is the one on the left is a BCL2 pathway, which controls the intrinsic apoptosis of cells, so whether a cell dies or lives with respect to some intrinsic cues. And if you dive into this particular network, for example, you realize that there's a lot of promiscuous interaction between components that displace each other, competitive oligomerization, and others. And the work on the right by the Elowitz lab on the BMP pathway is sort of highlighting a similar framework, which is there is a set of ligands, there's a set of receptors, and they interact with each other relatively promiscuously. And the exact combination of those leads to very specific outcomes. So I hope that by just showing this like very quick introduction to the field and sort of natural biology, there's sort of two frameworks, right? We have this sort of synthetic, mostly transcriptional biology where everything is orthogonal, sort of classic Boolean logic gate, layering, and the components don't cross those. And then we have this sort of natural, for example, protein signaling pathway, which I'm going to go into, which much more resemble this sort of artificial neural network architectures where you share weights, you have crosstalk, you have promiscuous interaction. And so without going any further in the introduction, I would like to introduce what the purpose of what my work has been is, asking the question is how could biomolecular computational capabilities and therefore engineering of life could be sort of supplemented by using de novo protein design. And specifically, if we could generate such protein networks for complex and compact logic. And as a first iteration of this idea, obviously biology uses phosphorylation and other events, but as a first iteration of this idea, I'm proposing here to generate just a protein equivalent to sort of a Boltzmann machine. In other words, a thermodynamic form of computation. So you have and in order to implement this, the design requirements are to have a set of dimers that can only form dimers, no higher order oligomers. So they need to share interfaces and be competitive with each other. We need to be able to have reversible dimerization between these different elements. And obviously we need to have a range of affinities and promiscuous interaction so that, if we have this all by all interaction matrix, this would map to a graph where the weights and the edges correspond to what we want to perform a specific computation. And then on top of this, obviously, the interaction of the protein in order to be detectable in a biochemical sense, each are either untagged, I'll call them untagged from now on, or one half of a split biochemical active component. So in all the work I'm going to show, it's luciferase. I'm just detecting luminescent as a proof of concept, but you can think of a split transcription factor or a split kinase or any other form of biochemistry that you can reconstitute upon reconstruction and actually function. And beyond just like trying to mimic a little bit more what biology has done, this also sort of goes into the idea of perhaps really tapping into sort of this information theoretic advantage of working with fewer components. And the little plot on the left is just obviously very simple and very crude, but if you think of increasing the number of species and you have a purely set of orthogonal components, the number of interaction you gain by adding species only scales linearly. And if you have a fully connected graph, that obviously scales with a power law. A fully connected graph will not be able to process information, but in that gray area here in the middle might be a sweet spot where we can extract and process a lot of information despite not having a lot of components. Now, this is all very theoretical, so let me dive into a specific example of what I mean by building such graphs. Oh, so first of all, sorry, before saying that, obviously it's a protein design project, so it's been leading on actually generating those protein components. The first iteration of these that I'm mostly going to rely on for the rest of my talk before moving on talking about more was parametrically designed. So we designed these sort of helical bundles. The The process is, what I described earlier, defining the geometry, defining the sequence, and then sort of tuning the interface in order to tune its biophysical properties and enable cross-reactivity, a range of affinities and others. Now, how do we implement an actual information processing, case? So here I've just taken one particular example. So if you wanted to compute the operation written on the left, A and not B or C, you have the Venn diagram and underneath the truth table for that operation. How would you go about doing this with a promiscuous network of interacting components? So here is a postulate of how, we can implement this. So you have a gray protein that is always present, irrespective. So that's the constitutive proteomer. And then you have the input proteomers, so the red, the green, and the blue dots, respectively A, B, and C. And so you can see and highlighted underneath, given the edges weight, which just means the strength of the interaction between these two proteins, that we see in, for example, the state 0, you have no signal. If you have only protein A, because it interacts with the gray one, the system turns on. And then if you move to, for example, the state 110, here we have the B component that more strongly interacts with the A component than the A component interacts with the gray one and displaces that dimer, therefore turning the system off. And then you can go through and you can see that by respecting exactly that graph topology, we can actually implement specifically this operation, which is the desired operation. Now, again, this is all on paper. How does it look in the wet lab? So the process is to make those proteins individually, to purify them, and then to assemble them. And the very early days of that project was, I would say, relatively crude in that I had a set of proteins that were purified in tubes. I was equipped with a pipette and some funky drawings that are made on, on the side to try to implement these logic gates and which components could be put together. And here are the results of 8 subgates. So, 3 input gates, 4 input gates, and then on the top is the logic operation, the Venn diagram, the graph topologies, and underneath is the in vitro result of that computation using purified proteins. And I'm not going to go the way I did earlier through all of them specifically, but hopefully what I can convince you with this, and I was very excited about it at the time because when I started this project, I wasn't really sure it would work, but hopefully what this convinces you of is, at least in some cases, it's not perfect, right? We don't have pure Boolean activation of the different states, but at least to a certain extent, we can get the right mapping, right? So the red squares are where the gate should be on, and then obviously the blue bars indicate the extent of the luciferase activity. And you can see that we can, these different graph topologies, if you choose the right one, you can actually map to a whole range of Boolean logical functions, which is very exciting because we can now really harness protein design and scale this up to go and explore much more computational, push the computational capabilities of such a framework. Now, this is only 8 gates. This was not that many proteins, and this was a lot of work and very labor intensive. So we needed to find a better way to sort of do that at scale. Sorry, before I talk about the moving on to at scale, I will talk about 2 things briefly, since my hypothesis was both that we could do this much faster than with gene circuits, and also the idea of compactness and the information theoretic advantage. So here is the demonstration for the gate reconfigurability. So here I just add the components in a stepwise fashion, and you can see on the right that, within sort of half hour or so, we can reconfigure the gate, suggesting that the system is obviously kinetically reversible and responds relatively fast, at least on the time scale that if we compared that to a gene circuit. And then the other thing I mentioned was obviously the idea of being more compact. And so on the left is sort of the state of the art of a protein-protein interaction-based, logic operation for a disjunctive normal form. And that required these 8 protein components to be made, and also making chain components, or larger species from a genetic perspective that may have undesirable biophysical properties, and then all of it. In contrast, the same operation here, I can implement it with using promiscuous interaction implemented with only 4 proteomers. So I think this really demonstrates this idea that we can achieve more compact computation with fewer components, which is really exciting if we're thinking of moving this into cells where genetic payloads are traditionally limiting. And so this really opens up a lot of possibilities. Now, I talked about the issues of not really being able to sample that much by hand. I mean, that's not unsurprising because I'm not that fast at pipetting. And if you think of just having 96 proteins and you want to assemble all possible combinations of 4, we're talking of almost 80 million possible combinations, right? So the space of these networks is enormous, and we want to find the ones that work properly. So what we've moved into more recently is, first of all, it's sort of semi-automated robotic pipeline to express and purify the protein at scale. So we can now purify sort of hundreds of proteins per week. And those are sort of the inputs to any network formation. And then the second stage, we also experimentally determine large all-by-all interaction maps so that we know the map underneath is actually experimental data of all-by-all 48 by 48 data points of such proteins, where we're trying to figure out what is promiscuous to what extent, and so what we should combine. And then we use acoustic liquid handling, which is extremely fast, that allows to do 10,000 transfers per hour. And so we assemble this in very high density microplates, let the system equilibrate thermodynamically, and then go in the plate reader and just measure the output. And so obviously for each gate, we have to sample all the possible states of the Boolean operation and ensure that each state is correctly either on or off as per the design. But that's sort of like rational design. But one first step that we can actually do is just to say, now that we have this capability of just exploring at scale, can we just randomly assemble with simple heuristics and see what the outcome is? And by heuristic, I mean, just pick out random proteins from your set, just ensuring that your assembled set contains, for example, at least a signaling pair or a signaling pair in one strong outcompetitor or something like this, right? So you can have relatively simple design rules, assemble them, and then look at the outcome. And this is exactly what this next slide shows. So, for the case of 4 input type of Boolean gate, you have 16 states and otherwise 65,000 possible gates. And for 5 inputs, it just scales massively to the billions. And what here it shows is by just randomly assemble with some simple heuristics, we can already sort of mine networks that map to operations. Not everything is perfect. You can see that blue region is things that just doesn't work. There is no signal whatsoever. Nothing seems to associate to others. And then you can see all the ones where you have yellow. You can have patterning that, if you can use, sort of Python packages to map back the logic operation. And you can see that indeed they map to specific operation. Some operations are more trivial than others, and therefore they occur more frequently. That's the case for example, these like large Smeary bands. This is just, A equals true, which is not very interesting in the case of a 4 input gate, but it is obviously one of the 65,000 possible cases. So this is sort of unconstrained, if you want, getting at making those gates. But we can also obviously more rationally think of, if we want a specific gate, what is the network that we need? So what edges do we want? And then go in that all-by-all matrix, look up the components, and then assemble the right thing. And this next slide illustrates exactly this. So this is sort of targeted sampling where, the gates were assembled based on pre-computed graphs that should map. And then as a verification, you can, reassign what the operation is. And this is just a 3 input, but reassign what the operation is and say, is it the correct Boolean operation? And again, you can see that the range of operation, in terms of just the set, is quite large. Again, a lot of trivial operation, A equals true, B equals true, not very interesting. Sets that rely on only 2 components like A or B. But you also have a whole set of operations that do actually make use of all 3 inputs, like A and C or B and C, etc. And I think, this is all, to me, extremely exciting because this framework really is demonstrating that it is working in order to generate these large, complex operations. And, the limit now is really just the computational primitives in order the proteins. And then one last thing, obviously, I'm sure many people will ask. So I have started working in collaboration with a colleague, Mohammed Abedi, on implementing this in, obviously, cellular context. Everything I've shown so far was in vitro. And it's very early days, but here we have arguably a simple operation, but we tried to implement this in mammalian cells using a reconstitution of a transcription factor. And, I think our preliminary results show that you have to play a little bit more around because it's harder to control concentrations in cells, etc. But you can, depending on how you permutate your transcriptional activated domain and the DNA binding domain, you can get to stages like the case on the right, where the Boolean function is actually recapitulated quite well. But as I mentioned, now we could hopefully go even further, and this is really enabled by recent advances in protein design, because of course, what I mentioned earlier, all these components have relatively small, little diversity from a structural perspective, and they were designed parametrically, were quite long, had higher failure rates, but we have all these new tools. And in particular, some tools that I have been involved in developing, and I think Alexi, my friend and colleague who spoke recently at the workshop, we worked together on defining a new framework using purely machine learning in order to generate the backbone and sort of do it in a generative way to sample higher structural diversity. And so we start from a simple prompt, the length of the proteomers, and in this case, the first target we were doing was just cyclic homoologomers. So I'm going to talk about heterologomers in a second, but cyclic homoologomers, and the prompt is just the length and the symmetry. We initialize the process. We go through this in a Monte Carlo way to try to optimize that sequence until it fulfills these requirements. And then in the second step, we use another network called Protein MPNN in order to design the sequence. And and how does that look is this on the left you have a spaghetti monster of not a protein, right? You would not order that, and you would not go in the lab to test it. And then through the optimization process, you can get, in this case, through a homopentamer that, at least by eye, looks very promising. And so the process seems to generate viable structures. And not only this, it generates a large diversity of these structures, which was extremely exciting for us because it really opens up this door of exploring the protein design landscape in a way that was just not permitted before because it was just too slow to do it with parametric mathematical approaches. This is a more formal way of saying what I just showed, which is like, the output of this network was very diverse in terms of the shape it could generate. Each purple dot here is an experimentally tested design that we went to the wet lab. And not only are they different to each other, they're also different to the training set of, in this case, we hallucinated with AlphaFold, the training set of AlphaFold, which was the PDB. And for people who are not aware, TM score of 1 means you match exactly the structure in the PDB, and the further to the left, the more different you are. But obviously, all of this was computational, but we did go on and validate these methodologies through crystallography. And I think Alexi already spoke about some of this, so I'm not going to delve into it too much. But suffice to say that this dual approach of using AlphaFold for generating backbones and then MPNN for generating sequences, so a purely non-energy based, but purely ML based design strategy really has yielded tremendous accuracy in how we could design those structures. So you can see overlaid the models and the crystal structures, and on the right is just the quantification in terms of RMSD. So you can really achieve that atomistic precision and having these more complex topologies. And I think Alexi showed exactly the slide. So he actually collected that data on cryo-EM, where we just expanded this process to larger structures and really showed that we could sample a space that nature had not, or at least the PDB does not contain, so the training set does not contain, and actually generate viable design. And so, I think this is now very exciting for my work and my interest in, generating de novo interactomes, because while before we had, this is the set, part of the set that I've been working with, you can see relatively little diversity in terms of what proteins we were working with. The one on the right is repurposing this algorithm to actually design heterodimers, and you can already see that the sort of shapes and types of protein we can generate is much more diverse, and that means we will have the ability to sample even more, hopefully, different ranges of affinities, ranges of promiscuity, to have a larger library of components that we can assemble into this protein network. Not only this, these new methods are exciting because they also allow you to not only design a single state, right? So everything I've talked about so far has been relatively straightforward. So we only have dimers, but we can do more than that. And obviously nature uses proteins in assemblies larger than just dimers. And specifically, we can also use that to do multi-state design using joint hallucinations. And this is just two examples where the sequence was optimized to be able to both adopt a homotetramer, which is the structure on the top in each case, and a heterodimer on the bottom. And the reason why I'm showing this particular case, right? I mean, the design framework enables you to design pretty much whatever you want. But the reason why this is particularly exciting is, again, for the purpose of molecular computation, and now that will be my last slide, because we can start to potentially directly encode complex transfer functions just from the assembly. So we don't need to have complex circuits. We can imagine that. And here what it is on the left and on the right are just thermodynamic simulation based on affinity between interfaces of a homotetramer and a heterodimer that are in competition with each other. And if you do a standard transfer function, a standard binding curve between just A plus B leads to AB, you have that classic sigmoidal gray shape. But now, instead of having A plus B, you have A4 plus B leads to AB, and you titrate the A4 component. What results as a consequence is this sort of log linear behavior in the transfer domain. And conversely, if you invert that system and you titrate just the B component, you get to almost sort of a digital, much sharper transition for going from 0 to 100% activation. So simple, in a sense, sort of concept of trying to harness stoichiometries, asymmetric stoichiometries, and complex assemblies can potentially lead to encode complex mathematical functions directly without needing much more bells and whistles to it. And I think with this, I will just conclude and provide a bit of outlook. So hopefully with this talk, I've shown that we are moving towards this hopefully post-evolutionary sort of world where populated by synthetic protein components. In particular, that these new ML-based protein design techniques really expand our ability to explore the protein design landscape, which is extremely exciting in terms of what it enables. And really, we can start moving, instead of just spending 5 years designing one component, designing hundreds of them and assembling them, in my particular interest case, into networks and systems for processing molecular information. And I think that also the results, although the validation itself is still pending for the more complex networks, but at least in vitro, I hopefully have demonstrated that these de novo interactomes do expand our ability to perform biomolecular computation in protein substrates with fewer components and faster. And so really that leaves the question of what are the limits of protein-based biomolecular computation. And also I've only thought about mapping all of these to Boolean operations, but of course we can start thinking about analog computing or generating classifiers where you don't only have the absence or the presence of a protein, but you directly compute its input in terms of the concentration dimension. And that's it. So I would like obviously to acknowledge a lot of people. The Baker Lab is a huge lab, and people say it takes a village, and that's definitely true to do a lot of this work. For the logic work, the mammalian work was done with Mohammed Abedi. The oligomer hallucination, my great co-workers, Lucas, Alexi, Rob, and Eustace. Also a collaboration with Chris Tashuk and Lancelot on doing these, pre-computing the network in order to map them in the wet lab. And then other people, and many people, to talk about some of the work I haven't been able to show today. And obviously, thank you to you and Jason for allowing me to speak today about my work. And I'm really happy to take any questions. Thanks.
Wow, that was wonderful. Thank you so much. A lot has happened since this workshop even. That's crazy. Well, thank you so much for presenting. I want to remind people of the algorithm. Usually I start asking questions, but you can stop me anytime by start asking your own. So just raise your hand here in the chat if you have any questions that you'd like to have answered. I would love to just perhaps if you don't mind, for you to go back a little bit more into detail on the slide before on some of the two last points that you had on there. Of just what are the limits of protein-based biomolecular computations and analog computing. Could you explain a little bit more, like, what could people be looking out for here if they were trying to make progress on these areas?
Yeah, so I think everything I showed so far is thermodynamic computation, but this is the simplest framework because it only relies on PPI, and it relies on the system solving itself. It's the partition function, right? Just computing that way. But nature is more complex than this in terms of obviously using non-equilibrium computation. And also, I didn't really have the time to go into this, but once you increase the size of these networks, it can become, you add noise and noise even more. And so what nature does obviously is amplification through catalytic steps, whether that's, for example, a phosphorylation event or, a gene transcription. And I think long-term, for the stability of these implementation in living systems, having amplification will probably be necessary. So that means that potentially you have small network components that compute a small operation that leads to an output that can be amplified by an enzyme, and then you can cascade this. And then in terms of analog computing, that's really just to say that, I think the framework from synthetic biology has been a lot on this idea of Boolean computation, which I have done as well here because it is tractable. There's a rich mathematical framework around it. But obviously, nature doesn't work in the Boolean domain exclusively. Some aspects maybe, but a lot of it is concentration dependent. How much ligand you may have on the cell surface receptor has a direct impact on its downstream effect. And so actually directly building networks that are working in the analog domain, so they directly take in concentrations of input and are part of the computation instead of just a yes, no, I think will be extremely powerful. Yeah, so I don't know if that answers your question, but that's sort of, I think looking forward, I think those will be very exciting areas to go into.
Wow. Okay. That's crazy. And I mean, I think we've, like many folks recently have been pretty affected by the bits that machine learning can do for their individual industries. I think the last year was really sped that up big time. And do you have any kind of either predictions on like what you may be able to do specifically with better machine learning tools like soon, or are there any plans, for example, to like in the year ahead to apply it in specific ways? Like what, can you give us any outlook on like how you're hoping to use better and better machine learning like in your work?
Yeah, so the work I showed on specifically the hallucination is, I think really on your first generation of generative quote unquote algorithm for sampling the protein landscape. There is two preprints that came out, one from our lab, one from Generate Bio, just before the holidays, that people applied and started experimentally demonstrating that pre-generative algorithms, such as diffusion models specifically, are extremely good at sampling the space. And I think, the Gen 2 of generative algorithm will be diffusion models because they faster, they really allow you to do more synthetic components. So I think that's definitely the future in terms of generative AI for protein design. The other aspect that I didn't talk about, and I think is extremely exciting, is the use of language models. I think you asked me the question on Twitter, but it was in a collaboration with Meta, who have implemented very large transformer-based language models to try to learn the deep patterns of sequence function relationships. We saw together with them that you can employ these to directly generate new protein sequences that work in the wet lab. And I think that's exciting because everything I talked about here is a structure-centric protein design approach, right? You define a problem geometrically, you design the shape, and then you try to make a sequence that goes through that state. And the reason to go via the structure is really just a proxy because we understand 3D spatial coordinates quite well as humans, but obviously biology doesn't specifically need the structure per se, right? I mean, the mapping is from a sequence to a function. And so if you can design for function directly, I mean, I think that's super exciting. How that will look like, I don't know. I think work by Neolia Peru, for example, who tries to make enzyme by just sampling this in a generative way from probability distribution that maps to enzyme classification is extremely exciting because you could imagine generating enzyme function by just sampling sequence space directly without having to do a theozyme and the small molecule binding, etc. And I don't know how that will go moving forward, but I think this is an exciting frontier for generative AI for biology.
Yeah, really cool that LLMs could potentially be useful in that way too. I know that people had tried to do something like that like 5 years ago or something, but it was really difficult. And now I feel like, we may have finally like crossed the crossed the threshold here. Okay, I know we have many hands up, so I'm going to stop asking for now. And Will, you go first, please.
Hi, great talk, Basile. I have a couple of questions. So one is on the technical side, in terms of how things are progressing, of what fraction of the sequences that you generate would you classify as well-behaved? So what fraction, well, how many sequences would you have to screen? Obviously, it depends on the function, but let's say in order to get pairwise interaction that you think is decent, how many sequences, what fraction of sequences would actually pass your threshold? And then the second question is kind of a more strategic one, which is that even though you could hallucinate a vast diversity of structures, if the goal is to generate a huge library of orthogonal interactions, then strategically it might make more sense to kind of adopt a common, one kind of boring structure, and then create some kind of modular combinatorial interface on that just might be more, might be easier strategically. So we can look at, for example, nucleic acids. They're very boring. They just make double helices, but you can make humongous combinatorial libraries of those very easily. Obviously, nature has used microRNAs and things like that are using base pairing. You can look at common scaffolds on enzymes. Enzymes you could say are very boring. They're all just alpha beta barrels or beta barrels and having loops on it. So is it actually going to be that useful to hallucinate all these structures if the goal is to make combinatorial orthogonal affinity matrices?
Well, thank you for your question. I'll ask I'll answer the first one first, and then hopefully not forget and move to the second one. So for the success rate, so for the heterodimers, that's still pending in terms of hallucination, because this is work in progress and I don't have wet lab data yet. This is only computational. For the original generation, which was energy-based design, which was most of what has led to the experimental work that I've shown, the success rate was not great, as in I think it was probably in the order of, well, I mean, it's probably 20%, which is not that bad, as in, you can order a five-fold excess of the sequences you'll need, and then you'll get that more or less. For the oligomers, which obviously is a different ballpark because there was the added complexity of symmetry, and it was obviously the success rate was symmetry dependent. Here we have a more systematic data set collection, which is published in the paper, where I think the success rate was the success rate was around 30% for secmode confirmation of the oligomeric state, which was very impressive to us at the time. So I think from a technical perspective, ML-based protein design has really changed the game in terms of being able to get to what we want, and faster with fewer iterations. Now, to your second question on, what is the advantage of the generative model? So first of all, I think because maybe there was a misunderstanding, you talked about getting orthogonal matrices. So actually, quite the opposite, right? I want to get promiscuous matrices, not orthogonal ones. The orthogonal one would be the classic approach of having this component just like chained together, but I want specifically promiscuous interactions, which at the moment I don't actually design for explicitly. So I only design cognate pairs just because multi-state design against end state is just not computationally tractable at the moment. So what we do instead is design the cognate pairs and then sort of dial in a set of properties at the interfaces that we hope will lead to more or less promiscuity, basically just simple biophysical heuristics such as the extent of hydrophobicity and things like this. I do agree with your point that it might not be necessary to generate these large sets, and I could focus on just a sort of a simple topology and sort of explore on this. But the advantage of sampling topology means that we get much more sequence diversity because there is obviously an enormous memory in the backbones when you want to design a sequence, and so I think it really allows to sample more. It might not be necessary. That is a fair point. I think where it may become interesting is once we would start to have, so everything at the moment is a single layered network, and so all the components are there. But if you start to think of what I answered just shortly after I finished, right, you would have a small network generating a computation that leads to that leads to another one, you would need to have separate networks that are all orthogonal to each other. In other words, you would need to have sub-matrices that are promiscuous within themselves, but orthogonal to each other in this sort of large-scale context. And I think in this context, it becomes interesting to have this generative AI approach, because you can imagine designing multiple class of topologies and then diversify within them. So I think that really offers a lot of opportunity going forward. I don't know if that answers your question, but
yeah, I think that's great. And yeah, installation is the really great motivation. And then also, if one wants to do the calculations beyond pairwise to make oligomers, then there the geometry comes back into play, and then the hallucination is going to be really useful. Great, thanks.
Thank you.
Awesome, thank you, Will. Next up we have Ted.
Oh, hi. Thanks for the talk. It's impressive to see this progress with machine learning to create molecular computation. So my question is about computation speed. So typically atoms are heavy compared to electrons. So you expect large molecule interactions to be relatively slow. And so I was wondering, to what extent can machine learning find faster equilibrating interactions, or maybe the dynamic non-equilibrium computation that you mentioned, as an additional criterion from just finding functional circuits in the first place?
So within this framework, at least, there is an inherent limit to the computation, which is the off rate and the lifetime of the complex. So it's not really the binding of a new component if your protein is isolated that is limiting, because unless there is major conformational rearrangement or others, the ballpark will be 10 to the 5, 10 to the 6 per molar per second, which at relatively high concentration, as one would expect them to be in a even in a nanomolar range, those will be over in a matter of seconds to minutes.
Mm-hmm.
The issue for the fast computation is obviously, if you do have a preset network, like in this case, right, after the step one, you have this timer and you want to outcompete it with the blue one, unless you have some active displacement, which at the moment this is not the case, you do rely on the lifetime of the complex, and that determines mostly the computational speed. And obviously, that would just mean, oh, well, then how about just making the lifetime very short? But that would also imply making very weak interactions, and then probably entering a regime where these networks are no longer really able to compute anything, because, if your interaction is 500 micromolar kg, you won't really have any population of that dimer. So I think, at least based on just PPI-based computation, the inherent limit will be probably in the order of the 30 minutes or so, just because of chaos. If there was a way to have an active displacement, sort of a, like what people have done with DNA, with toehold and others, that might change the game. I think this is probably something in the future, because as much as there's been huge improvement to making dimers, we're not there yet. It's not like designing an oligo where we guarantee the pairing and everything. There is a lot unknown about protein-protein interaction in terms of design. So yeah, but it's in short, I don't know how much that could be pushed. It feels to me that there will be an inherent limit based on the lifetime of the complexity.
Oh, okay, thanks. That's really helpful in pointing out where that limitation is. So that's great.
Awesome, thanks, Ted. We have Steve and then Aliyah.
Hi, thanks. This is really exciting on multiple levels. I just had a fundamental question, which is that obviously you have to be building these structures inside a cell using the environment and the mechanisms there, but can they then be exported and, if they're in obviously solvent, can they operate outside of a cell?
Yeah, so actually outside of this slide, so outside of the data that I'm showing here, where it was done in mammalian cell, all of that work, this and this, etc., all of this is in vitro. So the proteins are purified from E. coli, and then they're just in standard PDS solution. So most of the work actually and the data that I have so far is entirely in vitro computation. I think, if anything, the in vitro computation is simpler at the moment because there is a complete control of concentration. In the mammalian environment, or like the cellular environment in general, is more complex with that regard, especially using, in this case, transient transfection. Yeah, and then also the added difficulty, obviously, those proteins are de novo, so one would hope that they don't interact too much with other cellular components, but obviously, in the long run, there is also all the issue of having these networks that maybe work in a very purified setting, but once you put them in a cell, start interacting with endogenous components, and then that could lead to more fine-tuning or screening more network being a necessity for the long-term implementation of this framework.
Okay, thanks. A quick follow-up. What has been done as far as looking at variety of inputs and outputs? I mean, the forms at which you can, the mechanisms upon which you can read out, say, the answer of the logic computation.
Oh, so the readout so far, the only thing I have personally tested has been luciferase and transcription. So transcription being the data that is on this slide using a VP16 GAL4 system. I have not gone to other systems. I think Zibo Chen's work on with these orthogonal protein, I think they had gone to also yeast to hybrid, and it's escaping me, but I think those are the main components, maybe some other transcriptional systems mostly. Yeah, I think that's mostly it. One thing to keep in mind there is that the split component cannot bias the thermodynamics too much, right? Otherwise the computation goes astray. So actually that's the reason why I use luciferase and not split GFP, because evolution of the chromophore makes that interaction pretty much irreversible, and so your equilibrium shifts completely. So this system and the generalizability of it is predicated on having biochemical function that you can split, and where the split components are intrinsically not interacting with each other. But
So,
yeah, I've only tested 2 so far.
in engineering terms, you can end up with a latched output. You don't want that. Okay, thanks.
Pardon?
It's fine. Thank you.
Okay.
Wonderful. Okay, we're racing through questions here. Next one, we have Aliyah.
Hey, great talk. I was a bit late, so I might have missed this part if you talked about it. But have you thought about explicitly encoding negative design into the model itself? And this could be MPNN, so I'm talking more of like machine learning here. So it could avoid like undesired alternative structures, and this could be like edge-to-edge aggregation. So you can sample less of one class of motifs, just so you can work on cutting that noise down. I'm just wondering if you thought about that.
Yeah, so I have explored a little bit the multi-state approach. For the multi-state MPNN, I have not done it specifically for these heterodimers, but some other work in the lab, we've looked at it. I think the main issue is, for example, MPNN needs structure in order to be able to design against it. And once you have that, it does work pretty well, but you don't know a priori what the structure of the off-target state is. And so I think the multi-state negative design is an interesting approach in small setting once you maybe want to fine-tune or optimize a network. But I think at scale, it's difficult because the enumeration of all possible confirmation you may want to design against can be hard to achieve. And so I think from a purely practical standpoint, I think it's a very interesting approach, and I'd love to do it. But I think from a very practical standpoint, it is usually easier, in my experience, to sort of have implicit features of negative or positive design. So again, having more hydrogen bond networks at the interface versus less, etc. And then going in and experimentally characterizing how the edges and the extent of the cross-reactivity. That seems to work usually a little bit better, just because even if you could design and like on the computer, you would have the perfect case of a completely orthogonal interaction against that partner and a positive interaction with this one. You still don't know ultimately if that's going to be exactly the case in the wet lab, and so you still end up having to test it. So that's more from a practical standpoint, but scientifically, I agree with you. I think that would be a lot of fun.
Thank you.
Awesome, wonderful. Well, it's back to me in the last 4 minutes, and I would love to ask, like perhaps more of the longer term questions. And I know people don't like to speculate much, but with that caveat in place, what do you think could be possible in the next like 5 years, 7 years? I think 10 is perhaps a little bit too far out, but yeah, if everything goes right and if progress is continuing this way, do you have any like kind of carrots that people can get excited about when they monitor the space?
Yeah, so I will give a very speculative answer, because the extrapolation in the application domain is not quite my forte. I mean, this work for me was very exciting for its fundamental aspects. I think the one when I talk to people that, that comes up first is usually, adoptive cell therapies, for example, right? Where you could imagine a system where these proteins, because they are proteins, right? You could interface them directly with natural ligands. And so you could imagine having them for having more complex computation for a CAR T cell to enhance its safety or, lead to more complex sort of oncology profiles. I think that's one exciting aspect for sort of the biomedical application. Being more on the fundamental side, I think another exciting aspect of this is actually to design these for directly monitoring and probing cellular state and maybe understand more biology. Obviously there's been a tremendous advances in RNA-seq, proteomics, and other methods to try to quantify. Mostly seq method, right, to try to quantify a cellular state and then understand phenotypes and others. But it's more limited when it comes to proteomics being obviously the dominant method for high throughput characterization. But all of these, you need to have your cells and it's sort of destructive. What if you could have some sort of these networks that would integrate biomarkers and report on the cellular state, classify a cell, and then you could do live tracking, imaging, and really understand the state through which a cell may go over time in a live format. I think that would be a very exciting avenue for this application.
Yeah, very exciting indeed. And do you have any potential risks, challenges, collateral side effects or something that you think we should be monitoring as we go about this? Do you foresee any bits where you're okay, we should be careful over here?
You mean from a societal perspective?
Yeah, just from a scientific perspective. You see all the time with, LLMs also, potentially developing behavior that's a bit more deceptive, for example, if it has to be or something. Do you think, especially you having interacted with these models a bit, do you think there's any bits where you're oh, we should probably be a bit careful over here?
I think not more than what the same concerns are with GMOs and synthetic biology in general. I think this new technology, protein design, and its implementation for biomolecular computation at the protein level is just an enabling tool for working in that space, hopefully more efficiently and differently and complemented. But the same questions that have been since the inception of synthetic biology and gene engineering remain here, right? It's how are we going to responsibly apply these technologies when we start maybe implementing these systems in human cells or things like this? So yeah, I think all the concerns that are in the bioengineering space remain here, and I don't think it creates really new concerns outside of potentially accelerating how we could achieve these. But I think all the ethical dilemmas remain the way they were for synthetic biology as a field.
Got it. Thanks. Lots to discuss along the way then. Maybe the final question, I know we're at time. If people are now getting very excited about your work, in particular your work or the work of your lab and your collaborators, what could they do necessarily to help you along? It's a shameless plug moment. Anything, if you're hiring, if you're looking for funding, whatever it may be, now is a good time to make that clear.
So I think I can't speak. I'm a postdoc in the lab, so I'll speak by extrapolation to what I think what the lab is excited about and David is excited about is we have all these new tools, and I think the question is how do you use them and what do you use them for? So I think for specifically the Baker Lab, people that come from disciplines that are outside of the classic protein design and bringing a knowledge of either biology or materials and others or energy, etc., and having them implement their ideas with a new tool, which is protein design, I think is something that we're always very excited about, because we are now at an era where it no longer takes 5 years to get one molecule and then just prove that you can make it. You can actually make systems, you can make things that work better. So I think that's exciting.
Yeah, it's a great time to join the field. Well, thank you so much, Basile. This was really wonderful. Thanks everyone for joining, for asking such great questions. And I'll see you again here, I guess in a month from now, roughly. I think I already shared the workshop in the chat that we have going on this year. Thanks a ton. We'll be in contact with the video and the seminar summary and so forth. But this was really wonderful. And yeah, we're off to a great start of this year, I think. Thank you.
Yeah. Thank you very much.
Bye, everyone.
Have a good day.